the recording. And then we are aiming to post this to the to our website afterwards. So if you want to go back to it, you can. Let me get this. Okay. Great. And so are we recording? I can't see that.
Yes, I'm recording.
Okay, great. But welcome everybody. I'm Jessica Blois. I'm an associate professor in the chair of the Quantitative and Systems Biology Recruitment Committee. I'm joined by a variety of faculty and you'll hear about from later, as well as Fred Wolf, who is the chair of the QSB graduate group. And so we're here to sort of talk about the program and answer, give you an idea of the flavor of research that we do here at UC Merced and answer questions that you have about the program. And so as Fred wrote in the chat, but for those of you who are just joining, if you have questions, please try to maybe put them in the chat or use the raise hands feature and we'll get to you as we go through. Okay. Okay, let me go. So we're sort of done the welcome. So this is the plan for the day. I'm going to sort of go over the QSB program and talk about why we think we're sort of a fun, great program for biology flavored research. And then there's going to be 4 different vignettes on faculty research by the faculty that are listed there. And when they do the research, they'll each introduce themselves. Then we'll go back to a little bit about the QSB program structure once you're in the course here and in information about applying to QSB. And at the very end, we'll be joined by another professor who's going to talk about one particular program we have here called GRISE. So as I said, you can ask questions throughout, either probably in the chat is the easiest, but there'll be time at the end for you to have discussion too. Okay. So for those of you who are not familiar with QSB or UC Merced even, UC Merced is a relatively new campus. We are the 10th campus in the University of California system, and we're located here in the heart of the Central Valley. So there's a map on the left, and the star indicates the location of UC Merced. We opened in 2005, and we have a really dynamic and growing campus. So we're relatively junior as campuses go. We've had grad programs from the start, and I think QSB is one of the oldest, if not the oldest, graduate program at UC Merced. UC Merced is pretty centrally located in the state. For those of you who are not familiar with it, we're about 2 hours from the Bay Area, 2 hours from Yosemite. So here's some images of Yosemite from Yosemite National Park of Half Dome and such. But we're about 4 hours north of Los Angeles. Okay. So UC Merced is a really dynamic and growing campus. This image on the left is of one of the oldest buildings on campus, the Science and Engineering 1 building. That's actually where I'm located and seated right now, as are probably a few other faculty here. And we are rapidly expanding. So this image on the right is an overview of what campus looks like today. We've almost doubled in size and our physical footprint in the last few years, as we've sort of grown and expanded our enrollment and our physical footprint. And so now we have, let me just orient. So this building here is Science and Engineering 1, which is the one you see on the left. And then this whole part of campus on the other side of the canal is mostly brand new in the last 5 years or so. But, for those of us who have been here for longer, almost all of the campus is pretty new because we just opened in 2005, but it's a really dynamic, growing environment. There's beautiful lab space, beautiful office space and space to work and collaborate with your peers. QSB is one of the oldest graduate programs on campus. So it's the Quantitative and Systems Biology graduate program, and it's also the largest. So this is showing overall, we, as I said, PhD programs, master's programs have been, sort of part of the fabric of life at UC Merced since the very beginning. And we've really grown through time. So as of fall of 2022, we have about 779 students enrolled across campus. There's 38 students from 38 different countries that are represented. And I'm actually gonna, I'm gonna stop sharing for a minute. I have some other statistics and I arranged my screens the wrong way. So I need to get out of this to, go to a different view here so that I can show you those statistics. Here we go. This is better. Okay, are you seeing that same slide that you were before? Yeah, but I'm not. Oh, I see what I did. I'm not seeing my statistics. But QSB, how many grad students are there in QSB, Fred? About 124, I think, grad students this year in QSB.
Yeah, we're twice the size of the next biggest.
Yeah. And again, we have a number, I think we have students from at least 9 different countries represented, probably more than that, different countries represented. And so we're quite a dynamic grad group. Culturally, you can see we're quite large. And because of that, as you'll see, we have a really interdisciplinary environment. Okay, so what is QSB? QSB is the sort of shorthand that we use for quantitative and systems biology. We're a graduate program that offers foundational and cross-disciplinary training in biology to both doctoral and master's students. And our students are really coming to QSB from many different fields of science and engineering. Many students are coming, did their undergraduate degrees in biology or biology flavored degree programs, but we also get many students who are moving into biology research from engineering, bioengineering, from chemistry, from mathematics, etc. So our students come from a variety of different backgrounds. And then while they're here, because we're quite large and we serve almost all of biology, students are really learning or trained to communicate across the different disciplines within biology, as well as to our colleagues in our other graduate programs. But you're also then, training and gaining experience in core disciplines and skills that are needed to do your own research, right? And that usually is quite focused and quite specific to a particular area of biology. So across QSB, we have a number of different sort of areas of expertise, I would say. Probably the largest group of folks come from the molecular cellular biology. So that's, developmental biology, neurobiology, also microbiology and immunology down there. We also have a large group of folks who do work in evolution and ecology. That would sort of myself is included in that group. We have a subset of students and faculty who do research in biochemistry, biophysics, and bioengineering, computational and mathematical biology, and then a growing program in biology education research. So, yep, oh, here's the number. So, and so this is our faculty. QSB is a graduate program, and so the faculty in the graduate program, QSB, are housed or in different academic departments across campus. And so we have 124 grad students, as we said before, but we have about 68 faculty, and those faculty are associated with 8 different departments and 3 programs. So you can see that most of the faculty in QSB are coming from the molecular and cell biology department. That's this 38%. The next largest chunk of faculty are coming from life and environmental sciences department. That would be generally the ecology and evolutionary biology faculty, though not exclusively. And then the next sort of biggest chunk is bioengineering faculty. But we have faculty who are part of QSB who are in applied math, that's chemistry and chemical biology, I think computer and information sciences, let's see, material science and engineering and physics. And then we also have faculty from these three affiliated programs. So that would be the Joint Genome Institute here, Lawrence Livermore National Lab, and San Francisco State. And so we'll talk a bit about our partnerships with the labs in particular. And so similarly, the graduate students in QSB reflect that sort of diversity of faculty research, right? So most of the grad students in QSB are, affiliated or in labs whose faculty are in MCB. The next biggest chunk is life and environmental sciences, and then of course bioengineering after that. But you can see that students are advised by faculty in all of these different departments across campus. And so this is just a flavor of the diversity of the incoming class. We're not showing the whole QSB grad program because that would be too many people to put on one slide. But this is our incoming QSB class that sort of you can see, many people coming from various places near and far in California, in other parts of the US, as well as, various countries around the world. So, and I encourage you as you're thinking about applying to QSB to reach out to students and faculty as well, but to sort of get an idea of what the program's like from the student perspective. Okay, so why do we think QSB is great? I really like it, and I think because it really allows me to, and it allows students to study living systems across scales. And so depending on your particular flavor of research, some folks are more sort of theoretical or foundational, conceptual, and other students and faculty are doing research on the more applied end of things. So study might mean understand, explain, predict, or engineer living systems. But given the breadth of research that we do, some people are working with, particular genes. Other people are working with whole ecosystems. And so given this sort of breadth, you're really able to learn new concepts, new vocabularies, new skills that are cutting across all of biology. And so in QSB, we have a focus on that interdisciplinarity and sort of cross-discipline collaboration. We have a focus on quantitative training, as you'll see later in the talk when we talk about sort of the core classes that you will be taking while you're in QSB. And then again, through the research that each of you do, we're a research-based program that gives you that opportunity to really dive deep into a topic and master the concepts and technologies that you need to carry out your particular research. We have a number of different sort of partnerships or centers or facilities. This is listing just a few of them. So, as I alluded to, based on the faculty that participate in the QSB program, we have some pretty strong relationships with our national labs. And so, for example, we have this really awesome summer internship program with the Joint Genome Institute that many QSB graduate students have benefited from. It's been going on for summers for a number of years now. And so students are able to work with faculty or professors at the Joint Genome Institute and projects that are related to genomics, all sorts of different projects. We have a high performance computing cluster here, the CERT group, Cyber Infrastructure and Research Technologies group. We have a stem cell instrumentation foundry. We have a stable isotope ecosystem lab. These are all sort of core facilities that serve anybody doing research on campus. We have this UC natural reserve system for those who are doing more sort of ecologically themed research and many more. These are just sort of a sample of some of the centers and facilities and partnerships that we have. And we'll talk a bit later in the talk about how graduate students are supported in the program. But we, at this point in our life, we have a number of different training grants that support QSB students. Some of them are listed here: the Crest Center for Cellular and Biomolecular Machines, the Insight Biology Integration Institute, GRISE interdisciplinary program, and biomedical science and technology, and then this USDA Future Farmers program. I think we have a few faculty who are, participate in some of these different programs, and they can sort of speak to that as they're talking. And our students have been very successful. So this is just the recent awards that QSB students have earned. And so we have a few folks who have NSF GRFPs. We have folks that have different kinds of fellowships. So the Cognac Fellowship, which I think is supported, aimed at students who are coming from Mexico, I believe. The Royal Thai Government Fellowship, Fulbright, the HHMI Gilliam, UC, HBCU fellowships, et cetera. So a lot of our students are supported on fellowships and they're quite successful in their work here. And then once they get through the program and move on, our students are again, have moved on to a variety of different career, successful careers in a number of different fields. And so many of our students go on into academic positions. So we have QSB graduates who are now in postdocs or, some of them in tenure track positions. We have a number of former students who are working for the government labs, national labs, or state labs, for example. We have a number of students who have gone into industry or who are working with nonprofits. So sort of the whole spectrum of potential places one could go, our students are going to these different career tracks. Okay, so that's just a little bit of an introduction about the program overall. I wanted to switch to the faculty research talks, and maybe we'll start with Stefan. If you want to share, I will stop sharing. And then if you wanted to share your slides, introduce yourself and go from there.
Absolutely. Well, hello, everybody. Can you hear me okay?
Yes.
Yeah, okay, sweet, okay. So, I'm Stefan Materna. I'm a, third year assistant professor here at UC Merced. I'm, as Jessica already said, in the largest department that is contributing, that's part of QSB, which is the molecular cell biology department. And I'll give a rough overview of what my lab is up to and how I fit into the bigger picture here and how we collaborate across the department and across the graduate program. So I'm a systems developmental biologist, right? So I fundamentally believe in the interconnectedness of all things. That may be because I've read too many Douglas Adams novels when I was a teenager, but here goes. So what is developmental systems biology, right? So my organism of choice, you can see that already here in the background, right? It's the zebrafish. So on this slide, so there they are, about 3 days old or so. So that's what I'm interested in. And it's really the miracle of life. How do you go from a zygote to a fully formed organism? So this critter develops particularly fast. So you go from one cell to a beating heart in about 18 hours, right? Hard to beat. So the questions that we are asking, that my lab is asking, is really, how is the information encoded in the genome? How is that executed? How is that turned into an embryo, right? So you can rephrase that same question essentially, look at it from a different angle and ask, how is the behavior of individual cells really controlled during embryonic development? So when I'm talking about cell behavior, what am I talking about? All right, so let me just introduce you very concretely to the tissues that we're interested in. All right, so that's specifically the endoderm and the dorsiform cells, right? So I'm not going to go into the details here. So let's just say, when the embryo has about 1,000, 2,000 cells, so the embryo sits on the yolk over here. These cells sit at the margin of the embryo. Then the process called gastrulation, during which these germ layers are formed, ensues. And so these cells, which are very similar, so they express similar regulatory factors, they're exposed to similar signals, they do very different things. They both migrate, but in a very different mode. So the endoderm, which will eventually form the gut, they migrate as individual cells. They avoid each other and disperse across the embryo. That seems to be significant. Whereas the forerunner cells over here, they stick together and migrate as a group. So, and we're really asking, well, how can you start in such a similar place and diverse so quickly. So, seeing is believing. So I wanted to show you this movie. So we're now looking at the embryo from the right side here, right? So let me play this. So what we're seeing here at the bottom, that's the foreigner cells that migrate as a group. Up here, these are the endodermal cells, and you will see they try to stay away from each other. Then halfway through the video, they're converging, right? So let me show you this. There you go. So the foreigner cells, they disappear, the endoderm disperses, and now it's migrating together towards the midline, and that's forming the epithelium of the gut, right? So now we want to explain behavior of individual cells. Oops, let me stop this, move on. So what do? Well, we got to correlate gene expression in individual cells with that individual cell behavior. So, and we've started to do a whole bunch of single cell RNA sequencing. So where we enrich for these cells and then can draw a circle around these groups of cells that correspond to the foreigner cells of the endoderm here, and then find what genes are active, right? So we find, as one might expect, in the cells that move as a coherent group, they express a lot of adhesion molecules, junction proteins, and so forth, whereas the cells that migrate as individual cells, they express guidance cues and receptors. So that's the kind of stuff we do. But of course, that's not everything. So we're really spanning the whole spectrum from imaging, gene expression profiling. Then the organism we're working with really provides a wealth of tools for genetic manipulations. CRISPR is really just the tip of the iceberg here. And then single cell sequencing, we've already mentioned. Of course, we do a little bit of mathematical modeling to look into these behaviors. But of course, we're not doing this by ourselves. We're part of the QSP program here. And so there's a whole bunch of people that do similar things. I've listed a whole bunch of them here. So Stephanie Wu, I work most closely with her together, we're both zebrafish people. So then Fred, who's here on the call, he started doing single cell sequencing, as is Nestor, so we're coordinating on that. Susan is working on cilia biology and neurogenesis. And so what I didn't have time to mention is the foreigner cells are ciliated. So we have a whole bunch of proteins and genes that we're interested in together, and so forth. So here, there's the web address. Feel free to check out, what the people here on this slide do. And of course, we're not limited to this. So Jen and Nish, who will be talking next, so we're also collaborating on some single cell sequencing. So it's really a vibrant and very collaborative group. So I'll stop here then. Thank you.
Awesome. So if folks have questions for Stefan, maybe you could type them in the chat and we can sort of have a chat conversation. And then we can also come back to this at the very end. I'm going to pull up Nish's slides now and we'll have Nish go. I've fixed my slide issue. Okay, so are we seeing Nish's slides here?
Yep, and you can hear me, right? Everyone can hear me.
Yep.
So, hi everyone. My name is Michelle Nishiguchi, but everyone calls me Nish. And I'm a professor here in MCB, the molecular cell biology department. And we do marine symbioses in my lab. And so Jen, Jess, you can go next slide. And we work on a system between squid and their beneficial bacteria, which are in the genus Vibrio. They actually glow in the dark. And so my lab kind of looks at the balance between hosts and the microbes in terms of the evolution. So looking at the evolution of speciation between these bacteria, lots of different strain types, the ecology of what happens when they're not inside the squid, because this is an environmentally transmitted symbioses, and then any kind of altruism or conflict that occurs between the bacteria that are trying to get into the squid, because it's a great place to be. Also looking at the ecology, so what's happening outside of the symbioses. So this is environmentally transmitted, like I said, so the bacteria hanging around the water column for part of the time, and then they get back in the host. So that makes a big difference. And obviously abiotic and biotic factors that are going to be dependent upon driving that association, and whether there's a competition or cooperation between populations of getting the best symbiont. Next. So my lab does a lot of different, we kind of take it from the molecular all the way to the population level. So we look at kind of the natural and functional diversity of hosts and their symbionts. That's in the upper left corner. We sample, our closest sampling site is Hawaii. Our furthest one I'd say is probably Australia. And we do a lot of field work over in southern France. So there's lots of different places where we have a variety of questions we're asking about those types of populations, whether they're all living in the same place, which are sympatric, or across geographical barriers and they're allopatric. We look at the crosstalk between the bacteria. So that's the molecular signaling that occurs between the bacteria and the host. Oops, sorry. And then, and also that's related to, in this case, those are the genes that are regulating biofilm production, which is a symbiont characteristic that's necessary for colonization of the hosts. We do a lot of prediction of functional genes, and we like to manipulate things. So what we see in the field, we try to mimic it in the lab, but we also drive things in the lab by doing a lot of experimental evolution work. So we evolve different bacteria to different hosts, and we also evolve them in the test tube to say temperature increases or changes in pH, things that are relevant to asking bigger questions in terms of climate change. And more recently, we've been working with a lot of computational biologists on looking at gene network rewiring. So this is taking the transcriptomes and not just seeing what's getting upregulated or downregulated at one time, but taking all the different transcripts and seeing if there's any kind of pleiotropic effects between those, and not just within, say, maybe an ancestral state and an evolved state, but also between different strain types. So that's been kind of fun. I've been learning a lot of stuff about computational biology. So next slide. And so kind of we've been trying to combine this with doing some of our modeling. So this is looking at kind of, we call it ecoinformatics, kind of combining bioinformatics with the ecology of the system. So taking, looking at what's going on both spatially and temporally, say across seasons, looking at host microbiome diversity and abundance during those seasons, and then looking at like using proteomics or metabolomics to look at the functional diversity, and also then building these networks. And this is all happening in these squid vibrio symbioses. So a lot of different aspects. And like I said, my students kind of work on different parts of this puzzle, and all asking the kind of the overall question of why do these bacteria, are they so affiliated with these host squids? Why this particular squid and why these bacteria? So, okay, next slide. And I think, yeah, so I just want to make a quick announcement. So Jess actually announced that there's a few different kind of institutes or programs that grad students can apply to within QSB. So you apply to the QSB program, and we just got funded a big grant through the National Science Foundation through the Biology Integration Institute program. And ours here at Merced is called INSIGHT, which stands for the Institute for Symbiotic Interactions Under Climate Change. There's a bunch of researchers. A lot of us are in MCB, a few in Life and Environmental Sciences, a few in Applied Math, and one in Chem Biochem. So it's kind of multidisciplinary, interdepartmental, and we also have collaborators over at Michigan State and Resilient Oceans. And we're using 3 symbiotic systems here. The squid system I just talked about with myself and Elizabeth Heath Heckman at Michigan State. And then a sea anemone, Aptasia, which Maggie Sogan and MCB works on. And then the leafhopper symbioses, which Gordon Bennett over in LES works on. So we're using these models, and we're these 3 model systems to evaluate what's going on in respect to variations in climate change in the lab and in the field, and trying to model this using mathematical models. And then finally taking it to the conservation managers, saying, this is what we're predicting. These are the types of decisions you should be making, say, in Monterey Bay or wherever. So really trying to make this an applied effort to not only just get a better understanding of what's going on in these symbioses, but having a much more applied result from that. So we're very excited about that. We just lifted off on September 1st. And that's it. And so there's some information in my email, my website, and the BII's website. And if you have any questions, you can post them in the chat or email me or email Jess or Fred too. So
Great, thank you. Okay, let me stop share. I think it's Matt next.
Yeah, hi everyone. I'll share my slides from this side. So I'm Matt Hutchinson, and not officially actually a part of UC Merced yet, but I'll be starting in January in the Life and Environmental Sciences Department. And my lab focuses on large herbivores like the waterbuck that you see in this photo. And with these large herbivores, we try to understand how animals choose what to eat from their environments and how those decisions impact the individual animals themselves, as well as their populations and the ecosystems that they're a part of. So we do this work across a variety of systems, primarily in East African savannas, in Kenya and in Mozambique, where up to 25 to 30 different species of large herbivores coexist. And once we get to Merced, we're planning to start up new work locally in California, working closely with the California Department of Fish and Wildlife to understand how tule elk and pronghorn in the state move through their environments and choose what to eat. So at the level of herbivore individuals, types of questions we ask really center on how animals perceive their environment and how they choose what to eat from that. So this involves a lot of sample collection in the field, and by samples, I mean poop, which is a bit icky, but it's really a treasure trove from an ecological perspective, because we can extract and sequence the DNA within that poop to understand which plants herbivores were eating, and in combination with nutritional data about the individual plants, we can get a sense of how good a herbivore's diet was to make inferences about animal health, and also to test ecological theories about how animals select what to eat. At the level of herbivore populations, we're interested in how large herbivores and large mammals in general respond to environmental change. So the photos on the left show a before and after image of our study site in Mozambique when a tropical cyclone came through a few years ago. So you can see really extensive flooding in the lower picture. And right now we're trying to piece together evidence of how large mammals in that ecosystem responded. Using environmental disturbances like a tropical cyclone, we try to understand how different herbivores may have behavioral strategies to cope differentially with disturbance, how competition for food or for space changes as disturbance regimes change. And ultimately what we want to be able to do is to understand the likelihood that these large charismatic mammals can cope with and respond to extreme climatic events. Finally, at the level of entire ecosystems, we try to zoom out a bit from the plant herbivore dynamic to also consider other types of animals that rely on plants for food. They might be pollinators or fruit-eating birds. And using observational data about how each of these different types of animals rely on plants, we build models of the intergroup interactions going on, and then use experiments in the field in large-scale savanna experiments, as well as satellite imagery and the remote sensing data that we can get from that imagery to look at how ecosystem structure and the species interactions change based on which of these groups is dominant in the system. And ultimately, what we want to do with this work is to be able to advise ecosystem managers about how to best promote desirable ecosystem services. So if this kind of work sounds like something that you'd be interested in, please reach out to me by my email at the bottom, and you can also find out more on my website, either following the QR code or the URL. And yeah, I'd love to talk to you.
Awesome. Thank you so much, Matt. And same with deal, if you could, if you have questions for Matt, Matthew, you can put him in the chat there. And then finally, Dr. Jennifer Manile. Did you want to share your screen? Yeah.
I just have to find it, sorry.
It's like, I can share it for you, Jen.
Yeah, that might be helpful. I'm having some issues.
Yes.
Thank you.
Okay, let me do this.
All right, thank you. Thank you, Jessica. So hello, everyone. I'm Jennifer Manilai. I'm a professor in the molecular and cell biology department. And these are all my handles, although I haven't been checking Twitter recently for whatever reason. But you can contact me through any of these websites, LinkedIn, and also through Twitter. My research area is focused on the crosstalk between the skeletal and immune systems in mammalian models. So I consider myself a developmental immunologist, and my research projects require connecting elements in the fields of immunology, hematology, developmental biology, and stem cell biology. Here, next slide, please. Okay, so in my research, I've focused on the pluripotent hematopoietic stem cells in mice. These are the cells that are the source of all of your hematopoietic or blood cell lineages. And in the last years, I've been focusing mainly on B lymphocytes, which are the white blood cells that produce antibodies as part of your immune response. In mammals, hematopoietic stem cells reside primarily in the bone marrow of our long bones. So this includes our femurs and tibias, but you can also find hematopoietic stem cells in your hips, your ribs, your sternum, and calvaria, which is the skull bones. Next slide. Okay, and so within the last 15 years or so, work from many groups around the world have revealed that there is a dynamic microenvironment in the bone marrow where hematopoietic cell types interact with non-hematopoietic bone marrow stromal cells. And these interactions support hematopoietic stem cell self-renewal and the differentiation of these stem cells to specific lineages. So I have been focusing on the interactions between B cells and three types of non-hematopoietic cells in the bone marrow: mesenchymal stem cells, osteoblasts, and osteocytes. Next slide. So in general, this slide is just giving you a simpler presentation of what my research questions are. So I am asking questions that what directs a blood stem cell to become a specific cell type in the immune system? How are these immune cells activated? How do they recognize what is self versus foreign? And in recent years, I've become more interested in how immune cells are affected as we age, and also how bone disease can affect blood stem cell development and function. Next slide. So in my lab, we use a mixture of molecular, cellular, and techniques, as well as in vivo transplantation models in mice. And my lab relies heavily on flow cytometry and microscopy to answer our scientific questions. Okay, so just to give you some context of how I fell into this work, I just wanted to share with you a short story, and starts with osteoporosis. So most of you may know that osteoporosis is a disease that's often acquired with aging. This results in weak and brittle bones that can lead to a risk of bone fracture. Osteoporosis is a problem worldwide, and it's a major health issue for the aged. And most medications to control osteoporosis focus on inhibiting bone resorption by a cell type called osteoclasts. And it's only recently that new drugs that focus on the opposite effects, that focus on bone building, have become available. Next slide. Okay, so one of these new bone building drugs is called sclerostin antibody. And sclerostin antibody is basically an antibody that will deplete sclerostin levels in vivo. And these antibodies have completed clinical trials, and they've been shown to be very successful in increasing bone density in osteoporosis patients and reducing future risks of fracture. So if you look at the image on this slide, it's called a micro CT. These is from femurs of the bones of saline treated mice, so controls, and mice treated with anti-sclerostin antibody on the right. And if you look at these scans, you can see that there is a significant increase in the bone formation within the bone marrow cavity. And that is the site of hematopoiesis, where my research interests lie. So I have been studying the effects of reducing sclerostin on the immune system in mouse models for quite some time. And so next slide. And so this is just a quick summary of the findings from my lab. These are all published and available with open access, so you can follow up if you'd like to read more. But since 2012, my lab, which has been mostly graduate students in the QSB program, have published several studies examining the role of sclerostin deletion in immune cell development. Our first study identified apoptosis in B cells in the bone marrow of sclerostin knockout mice. And we also found that this B cell defect was related to decreased levels of a B cell growth factor in the stromal cells of the knockout mice. So this led us to conclude that the bone marrow microenvironment was altered due to changes in bone structure after treatment, or sorry, after deletion of sclerostin. We then followed up the study with an analysis in conditional sclerostin knockout mice, where we deleted the sclerostin gene in mesenchymal stem cells, osteoblasts, or osteocytes specifically. And this analysis surprisingly revealed that sclerostin deletion at the mesenchymal stem cell stage and the osteoblast stages resulted in B cell development defects, but that deletion at the osteocyte stage, the latest stage of bone cell development, did not affect B cell development. Okay, and then in our most recent paper, we studied older sclerostin knockout mice, and this analysis revealed hematopoietic defects in the knockout, including evidence of inflammation in the bone marrow. So taken together, these papers and our studies have shown that sclerostin is a new mediator of crosstalk between skeletal and immune systems, and this to our knowledge, are the only reports that show the effect of sclerostin depletion on immune cell development, and suggest that osteoporosis patients that are receiving these new drugs may have problems with their immune cells. And so we are currently seeking new funding to follow up on this in humans. So next slide is the last one.
Okay.
Yeah.
I was like, we need to move on.
Yes, I know. Okay. Oh, sorry. Next one. Okay. So just to summarize, because we have to move on, I have another bunch of projects that are working on studying the effects of changes in bone development on immune cells. And we've been developing new mouse models, conditional mouse models, to delete other genes that have similar effects. And so if you're interested in learning more, you can contact me, and I hope to see you all in Merced very soon. Thank you.
Awesome. Okay, let me switch presentations here. Okay. And share my screen. Okay, so let me go on. So that's a little flavor of the breadth of research in QSB, right? You can see we span from cells and genes to whole ecosystems, and I think you got a little sampling of that just in these 4 talks. Once you're in the program, we provide a lot of support for students throughout their career, right? Each of you is going to be developing your own research. And so typically you'll be supported in that by, through your advisor. And so every student will have at least one advisor. Some students are co-advised, and these folks are, people like myself or Fred or Nish or Matt, who are there to help advise you in research directly in your area. They're your main contact. There's a number of other support systems that help you get the sort of depth and breadth and support you need throughout your program. Every student has a thesis committee that helps guide their work across their academic trajectory. And then we're, of course, part of the larger graduate division. So people like Jessica Ventura, who's on the call, and others are helping support students throughout their career. This is what sort of a typical sort of plan in QSB looks like, right? Your first couple of years, you're spent doing lab rotations, which we'll talk about a little bit next in coursework. By the end of your second year, we aim for students to have developed and defended a proposal and go through the oral qualifying exam so that they advance to candidacy. And our goal is that all students are finishing their PhDs by the end of their fifth year. Typically, let's see, are you seeing? Yeah, typically the time to PhD is about 5 to 6 years. So the median time right now is about 5.5 years for students finishing their PhDs. Okay, so as I think this came up in the chat, in year one, students often, and this is really encouraged, are doing rotations. So we have sort of 2 tracks in year one. So most students are going into the rotation track, where they do rotations amongst at least 3 different faculty members or 3 different labs within QSB in their first year. And then at the end of the year, you choose by mutual agreement with the advisor which lab you're going to join. And this really provides students a flavor of what the research in the lab is like, what the community's like, what it's like to be in that particular lab, and it gives you a good sense of whether that research that you would do in that lab is where you want to take your career. A subset of students come into QSB through what we call the declaration track, where they have a very clear idea of what they want their future research to be. They know exactly who they're going to work with, and so they apply to work directly with a particular advisor or co-advisors. But most students are really encouraged to go into the rotation system. In your first couple of years, you're also doing a variety of coursework, taking a variety of courses. And so the core QSB requirements are that every student needs to take one class that fulfills the systems biology requirement and one class that fulfills the quantitative biology requirement. And then beyond that, you'll take an elective. All students need to do attend our seminar series in their first year and take an ethics class. And then you're often rounding out your degree, your coursework with other courses as needed for your particular research topics. We do have 2 different and a third soon optional concentrations. And so we have you can get a degree in quantitative and systems biology with a concentration in molecular and cell biology, or in ecology and evolutionary biology, or this developing one, it's under review right now, in computational and theoretical biology. Okay, so the important stuff, applying to the program. The priority deadline for the QSB program is December 15th of this year. And so we really encourage everybody to get their application to the program in by that date. What does it mean to be a priority deadline? It means that you are first in line for consideration for funding, right? So there's a variety of different internal fellowship opportunities. Those have pretty early deadlines, and so we need some time to evaluate applicants. And so the students that get their applications in by the priority deadline are the ones who are guaranteed to have their applications considered for these internal fellowships. The final deadline for applying, though, is January 15th of next year. And there's fee waivers available for folks who attend these recruitment events. Everybody here, Jess Ventura will be emailing you with the fee waiver, or you can also just email myself at the QSB inquiries at UC Merced with your request. As I said, when you initially apply, you need to choose whether you are applying through the rotation track or declaration track. And then once you're admitted and you decide to attend, at that point, then you choose your specific lab. That comes later. We use a holistic admissions process. So every student needs to develop a statement of purpose. You also will develop a statement of personal history and achievement contributions. Every student needs a number of letters of recommendations, and then GREs are not required for our program. And we're using this application package to assess for two purposes. The first is to assess your suitability for admission to the program, but we're also using this to assess your suitability for particular internal fellowships. And so we encourage you to really, think deeply and prepare well for all parts of those application, and in particular, both the statement of purpose, which is where you really focus on your research interests and experience, as well as your personal history and achievements contributions sections. This also came up in the chat, but regardless of which track, whether you're going rotation track or declaration track, we encourage you to contact faculty as well as other students who are in the program to see, whether your research interests find a good fit in QSB. If you are in correspondence with particular faculty, you can mention them in your application. You can check their names, I think, somewhere in the application package. And then again, it's always good just to ask your letter writers if they can write you a strong letter of recommendation. Students are supported in a number of ways when they're here. So we usually have, I think all students are guaranteed 5 years of support while they're in the QSB program. That support has a number of different flavors. Some students are on research assistantships or GSRs. Those are typically grants that your lab has that are made available for you to work on projects associated with your advisor's research. A number of students are supported or on teaching assistantships. And then a number of students also have either internal or external fellowships, such as the NSF GRFP or UC Chancellor's Fellowship, et cetera. So when you're in the program, your typical annual salary right now is about $30,000 per year. You also have the tuition and most of your fees covered. And so you don't have to pay anything for tuition and fees. And then QSB also makes available, we have a fund to support conference travel for students, as well as your labs often have, through your advisor research grants, have support for travel to conferences to present your research. So there's a number of different ways to support your work here. And then this is just a final slide, looking at the affordability of Merced compared to our peer institutions, our other UC institutions. We are one of the more affordable cities to live in California, and amongst the UC institutions, the cost of housing here is substantially less than in other places in California. And so so our wages actually, help, match the cost of housing better here than in other places. I think I saw Professor Ajay Gopinathan come. Yes.
Yes.
And so we finally, just in the last couple minutes, maybe just 1 minute, couple minutes, Ajay, if you could talk about the GWISE program. This is, again, similar to Insight, one of the programs that students in QSB can apply to join, I think. But.
Right, definitely. Thank you, Jessica. Thanks for being flexible. So yeah, and just this is a National Institute of Health funded graduate training program. So it's really a fellowship that will support you for your first 2 years. And so if we go to the next slide, I can just tell you a couple of things about what iBioSTEP is. It's an interdisciplinary biomedical science and technology program, and its basic idea is to prepare a diverse pool of scientists earning a PhD essentially in careers in the biomedical field. And our main emphasis is the interface, or the kind of the frontiers of the intersection of physical sciences, engineering, and biology. And the idea is to bring physical science engineering perspectives to understanding biological problems. And this is open to first-year students in 6 different graduate groups. So people from all these graduate groups are admitted into the program, and they form a cohort, and each cohort is about 6 to 8 students, depending on how funding goes. And there's 2 years, so there's 12 to 14 of you at any given time. And if you go to the next slide, I can just tell you a little bit about kind of the activities and the timeline and why you should actually be in this program. And so one of the things, that's a type, it should be like 30K, but it's a stipend that's commensurate with other fellowships on campus. We also, and tuition and fees per year for the first 2 years. But we also have additional travel support. So you can actually go to conferences and workshops. And most importantly, through the program, we have multiple opportunities for professional and career development. So we host a number of workshops on literature survey, enhancing rigor in your research, public speaking, things like that, writing papers. We also have courses that we offer through our program that you can take as electives in your individual graduate groups. So for example, QSP allows electives that you can take. And then we have different career tracks. And I can share the slides with you later. You can look through the kind of timeline of courses and activities throughout. And that's the last slide. I think if you go to the next slide, it basically tells you how to apply. You go through the normal application procedure, just like you would apply to QSB. And in one place, you'll see a thing that says, are you interested in the GRISE program? And if you are, and a lot of QSB students are, just click yes. And then we ask for a little blurb to tell us why you are interested in this program, what it is that makes you excited about it. And it's a very competitive program. We get about 80 applicants a year. So, feel free to learn more about the program, reach out to me or any faculty that are in the program that you're interested in, and be happy to chat with you. So that's it. I'll stop there. Thanks
Great.
again.
Thank you so much, Ajay. Okay, so that is the last, we probably had one more slide, which was just said, if you have questions, what questions you have.
I'll put my chat email in the chat too.
Yeah. But I see that there's a number of questions in the chat. But if anybody has some time, and if you need to leave, we're getting close to the end of the hour, so feel free to drop off. But if you have questions, maybe raise your hand or unmute yourself and type them in the chat. Yeah, Marisol.
Hi, everyone. My name is Marisol Contreras. I'm a first-gen college graduate from UC Davis, working in biotech. And so my interest is going towards the QSB program because I love genetics and I find it very important for understanding the differences between drug dosage and stuff like that. And so I really want to know a little bit more about kind of the translational research possibilities outside of the basic research for some of the labs, how much clinical data is available, and kind of is that resource commonly available for all UCs? Kind of just give me a little insight to the possibilities there, please.
Let's see. Yeah, I don't know. Other folks can chime in. In terms of the translational research, the folks that come to mind are some of our folks like Kara McCloskey or some of the folks in bioengineering who are doing some of the more applied work. Access to clinical data, there's not a UC-wide standard for that, right? That access to clinical data is going to be based on the particular research data you need to answer your questions. And so I don't know, I'm not quite sure I don't know if that answers your question. Fred, Jen, other folks maybe in the MCB that do more that translational science step?
It would depend really on the advisor that you're working with, what type of information they work with and what's available. So I recommend just getting in contact with a variety of people whose area of research might interest you and talk to them about it.
Yeah, Francisco.
Hello, everyone. My name is Francisco. I'm from Mexico. I am intending to apply to the program. And my question is about the application. I have 2 degrees, 1 degree in biology. I am reaching now a 2nd degree in biotechnology engineering. But unfortunately, I think I don't get my translations from the 2nd degree on time. Could I just apply with biology? I just mentioned that I am finishing a quantitative degree in biotechnology.
Yeah, I think that I mean, I think you can in the application form, right, if it has list your degree, and I think probably for international students, when you submit transcripts, there might be some like transcript translation that needs to occur. If you have that already done for one of your degrees, that's fine, do that. You can also talk about your overall pathway in your both of your degrees, either in your research statement, you know, in that history that you have informs maybe where you want to go with your work next, or in your personal history statement, right? Or both, right? So you can certainly talk about both of your degrees, and don't let the fact that you have not finished one of them completely or not gotten it quite translated completely limit you. I would certainly mention that because both feel relevant here. Yeah. Other questions? Yeah, Francisco, another one.
Again, I know there is a special fellowship from the University of California and Mexico.
Yeah.
Recently, I have checked in the webpage, and unfortunately, they have problems with the administration, and I think they pay for the application, but I can use the free wave that are you saying for that purpose?
Yeah, I believe that you'll be emailed with the fee waiver. So all attendees for this recruitment webinar get a fee waiver, and that applies for international or national students. And so that will waive your application fees. And then once you're here, yeah, there's this Alianza program, right? It used to be called UC Mexis, and a number of students, I don't, you know, I'm not quite sure where it is, whether they're administering grants right now, but that is a program that has supported UC Merced students in the past. Yeah, Prince. You're on mute. Yep. Prince, you're muted.
Oh, sorry about that. Okay, so I'm Prince from Ghana. Read a lot about the QSB program, and I think I'm very interested. But I want to ask, I'm currently working in a research environment where I'm working on phase 3 clinical trials for malaria, because malaria is an endemic infection in Africa. I want to know, I don't have a research publication yet. How does that affect my application to the QSB program?
It's like, I don't know if anybody of faculty here have been on the QSB admissions committee, but I guess that the majority of students that apply to QSB do not have publications already. So, you know, really I think at this point, it's talking about your interests, your research interests, where you want to go, maybe any past experience you have. If you have experience in a research environment, certainly talk about that, mention that, and how that sort of has shaped your interests going forward. But certainly apply. Don't let the lack of a publication hinder you. That is totally normal.
Yeah, I'll jump in for a second. We have a very holistic view of the applications. We look at the letters, we look at what you write, we look at your education. If you have publications, wonderful, but if you don't, it doesn't matter. It can be supported by other aspects of your application. So we really look at the person as a whole as much as we possibly can.
Marcel.
Can we please talk about the collaboration between other institutions as well? I actually live in San Francisco, and so I'm from the area, but I'm also have family in Merced, so I'm very interested in both locations. I wanted to know if there was any like co-mentorship, or just know who's on the Bay Area side, and you know, how can I collaborate with both? I care about both communities.
Yeah, I mean, I think that the biggest way we see this diversity or collaboration is, I mean, there's two possible ways, right? There's sort of through the research you're doing, if you're doing it in collaboration with researchers at other institutions. But many students in QSB have, every student has to have a thesis committee. And so many people have committee members who are not from UC Merced. I would say, that's relatively common. And so if you have someone you've been working with at an institution in the Bay Area or elsewhere, you can invite them to be on your thesis committee. We do have, one of the faculty within the MCB department is a faculty member at San Francisco State, but has an adjunct position at UC Merced in the MCB department. And then we have a number of faculty from Lawrence Livermore National Lab, Joint Genome Institute, who participate in the QSB program, primarily in the genomic space. And so in that case, in some cases, they are either primary or co-advisors for students. In other cases, they're committee members on students' theses.
Okay, and can you give me the name, or maybe I'll email regarding the name of the Genome Institute faculty member?
If you look on, if you look at QSB, the website, and look at our faculty, it's there's a Zhang Wang and Axel Wiesel, I think, are the two faculty members at JGI.
Okay, John Wayne, you said?
Yeah, it's Fred just put that down in the chat.
Thank you so much. Thank you.
And Fred, I'm gonna have to hop off to go to a different meeting, but if you could take over as host, or I don't know.
So I think we have to finish up now.
So we will have to finish up now. Yep. And so please feel free to, thank you everybody. Thank you to the faculty for attending and presenting your work. Thank you to students, prospective students for attending. If you have any questions, you can email QSB inquiries. You can email myself or Fred or any of the faculty on this talk or in QSB directly, and we're happy to answer your questions. So, okay.
Awesome. Thank you, everybody.
Thank you.